Good day, everyone. So for this lesson, we will be talking about mechanisms of fibrinolysis. So we already talked about the primary hemostasis, secondary hemostasis, and then the last stage would be the fibrinolysis. Okay? So we will be explaining the terms related to fibrinolysis and its mechanisms, and then discuss its importance. Okay, so to start, okay, so what is your fibrinolysis? So fibrinolysis is a complex process that controls both hemostasis and thrombosis. So this is a physiologic process that includes gradual or progressive removal of fibrin, cleaving it into soluble fragments, which is then removed by your phagocytic cells. Okay? So as we already mentioned, di ba, once the tissue or the area na nangangailangan ng clot is already healed, okay, so hindi naman pwedeng doon na lang forever si clot. Okay, so ang purpose naman now ni fibrinolysis is to take out the clot, dissolve the clot para bumalik na yung dating function or yung dating flow of blood within that area. Okay? So main objective, remove the unwanted clot to restore the normal blood flow, as I've said, on the area to facilitate the repair of the damaged blood vessel. Okay? So the regulation of your fibrinolytic system is mediated through a wide array of cofactors and inhibitors that maintain the hemostatic balance in a delicate equilibrium. Same with the other stages, okay, so meron din tayong activators, meron ding mga inhibitors. Okay. So this again is your last stage of coagulation. Okay? So responsible here, ang nangyayari here is you have digestion of fibrin clot, okay, that keeps the vascular system free of deposited fibrin or fibrin clot. Okay? So fibrinolysis begins with the activation of plasminogen to plasmin. Okay? So ang pinaka-important natin ditong enzyme is your plasmin. Okay? I believe it's already mentioned on the earlier discussions natin. Okay. So this is a brief overview ng fibrinolytic activity. So we have here our fibrin. Okay? So once done with the yung common pathway, di ba, our end goal is to have a stable fibrin clot. Okay? So our fibrin clot is here. Okay? It will be dissolved in the presence of plasmin. Okay, saan magaling si plasmin? Galing siya kay plasminogen. Okay? So plasminogen is present within your blood. It will be converted to plasmin in the presence of plasminogen activators. Okay? Yan. So how does your body regulate the fibrinolytic activity? So you have different types of inhibitors. Meron tayong plasminogen activator inhibitor. So in this stage pa lang, it will stop the process na. We have what we call plasmin inhibitor. Okay, so this one naman, so ito yung ini-stop niyang process. Okay, so it neutralizes the effect of the plasmin. And meron din tayong tinatawag na TAFI. So si TAFI naman, okay, ang work niya is to work on the fibrin. Okay, so si fibrin yung ini-stop niya. So si fibrin po yung naapektuhan. So even though there is presence of plasmin, kapag meron kang TAFI, it won't be degraded. Okay, so this is also another type of inhibitor. Okay. So what are the different proteins po in your fibrinolysis pathway? So again, we have plasminogen. Okay. Plasminogen, okay. Ang active form niya would be plasmin. Okay, so it is a serine protease which digests your fibrin clot. We have tissue plasminogen activator. Okay. So this is also a serine protease secreted by activated endothelium, which activates your plasminogen. Okay? Galing sa endothelial cells as well. Okay? Kaya, di ba po, hindi po, ang inyong hemostasis activity, hindi siya step-by-step process. Almost simultaneously, naglalaban po yung inyong procoagulants and anticoagulants and also yung inyong pro-fibrinolytic enzymes and not anti-fibrinolytic enzymes. Okay? So, kaya it's a balance. Dapat may balance. We have urokinase plasminogen activator. This is also a serine proteases secreted by the kidney cells. Also activates plasminogen. Plasminogen activator inhibitor. This one naman is yung it inhibits tissue plasminogen activator. Okay? Antiplasmin, it inhibits free plasmin. And then thrombin activatable fibrinolysis inhibitor or the TAFI, which suppresses fibrinolysis by removing the fibrin C-terminal lysine binding sites, blocking the TPA and plasminogen binding. Okay? So let's we will discuss it further. Okay. Again, plasminogen. So plasminogen is a circulating inactive protein. So it is a glycoprotein synthesized in your liver. So it is freely it is a protein po found in your blood. Okay. However, it is also stored and transported in eosinophils. Okay. So increased concentration associated with inflammation. Okay. So si plasminogen po, wala yung function unless it will be activated or converted into plasmin. Okay. So again, plasmin is a serine protease that digests your fibrin clot. Okay. So this is the active form of plasminogen. So an enzyme responsible for clot digestion. Okay. This can destroy fibrin, and factors 5 and 8 as well. So, hindi lang siya nag-work sa fibrin, it also works on factor 5 and factor 8. Okay, so factor 5 we know is on the common pathway. Okay, involved sa common pathway. If wala kang factor 5, hindi na rin mabubuo ang clot mo talaga. Okay? Okay. Okay. So, how does plasminogen get activated or converted to plasmin? So we have again what we call the tissue plasminogen activator. So this is from the endothelial cell. Okay? So once po damage si endothelial cells, so hindi lang procoagulants yung kanyang sinisecrete, it also secretes fibrinolytic enzymes. Okay? So this hydrolyzes the fibrin-bound plasminogen. So circulating tissue plasminogen activator is bound to inhibitors such as plasminogen activator inhibitor 1. Okay? So, ito, one of the balancing mechanisms. So, si TPA tissue plasminogen activator, we have what we call a circulating TPA. So, aside from the epithelial cell, we can also find it sa blood. Okay? However, it is bound or nakadikit siya sa inhibitor, kaya hindi siya mag-work basta-basta on activating the plasminogen. Okay? Okay? So, some medications po na pangtanggal ng blood clot, pantunaw ng blood clot, they mimic the activity of tissue plasminogen activator. Next, we have urokinase plasminogen activator. So this one naman is secreted by urinary tract epithelial cells, your monocytes, and macrophages. Okay? Circulates in our body at a value ng 2 to 4 ng per ml. Okay? This one also hydrolyzes fibrin-bound plasminogen. Okay? So, kaibahan ni urokinase ay sa kay tissue plasminogen activator. So this one does not bind firmly to fibrin and has a relatively minor physiologic effect unlike your TPA. Next, we have streptokinase. So, streptokinase, this one is comes from as a product of beta hemolytic streptococci. So, in cases of you have streptococcal infections, okay, so it can be a factor po to stimulate the fibrinolytic activity. Okay? So your urokinase, it directly activates your plasminogen into plasmin, di ba? And your streptokinase, it forms a streptokinase plasminogen complex. Okay? It forms a streptokinase plasminogen complex, which then converts plasminogen into plasmin. So again, plasminogen is secreted by your endothelial cells, diba? And then it will be converted into plasmin. However, we can also see free-floating plasmin in our circulation. Okay? So itong free-floating plasmin na to, it has the ability to digest fibrin and fibrinogen. Okay? So however, we have a regulation or balancing mechanism. Okay? So, ini-neutralize po ni anti-plasmin si free-floating plasmin. Okay? So, your anti-plasmin rapidly binds and inactivates any free plasmin. Okay? So, we have a condition what we call primary fibrinolysis. Okay? So, primary fibrinolysis happens when the free plasmin is left unchecked. Okay? So, hindi po nano-neutralize or it's not enough yung free floating plasmin kaya nagkakaroon ng fibrinolytic activity kahit na walang clot. Okay? So, plasmin is formed in the absence of coagulation activation and clot formation. Okay? So, this is a dangerous condition. Okay, so it can cause a potentially fetal hemorrhagic outcome. Next, we have what we call secondary fibrinolysis. Primary fibrinolysis naman is because of the breakdown po of blood clots due to a medical disorder or medicine or other causes. Moving on with the inhibitors. Next, we have, first we have plasminogen activator inhibitor. So this is the major inhibitor of your tissue plasminogen activator and your urokinase plasminogen activator. Okay, also secreted by endothelial cell, the megakaryocytes, smooth muscle cells, and fibroblasts, monocytes, adipocytes, hepatocytes, and other cells type. Okay. We have A2 antiplasmin. So this is the primary inhibitor of plasmin, which we have mentioned kanina. Is synthesized in the liver. This cross-linked into the fibrin clot by activated factor 13, so renders the clot resistant to fibrinolysis. Thrombin activatable fibrinolysis inhibitor. So this is also synthesized in your liver, activated by the thrombin thrombomodulin complex. So it inhibits fibrinolysis by cleaving exposed carboxy terminal lysine residue from partially degraded fibrin. Okay, so once na ginawa to ni Taffy, so magiging resistant na yung inyong fibrin to plasmin. So hindi na siya mag-degrade. So, we keep on saying na ang goal ng fibrinolysis is to degrade the fibrin clot. So, what are the fibrin degradation products? So, anong nangyayari sa fibrin? Okay, once nangyari to. Okay? So plasmin degrades fibrin in the clot and also fibrinogen in the plasma, forming fibrin or fibrinogen split degradation products or FDPs. Okay? So we have this type, so different types ng fibrin fragments. We have X, Y, D, E, and DD, or the D dimer. Okay? So D dimer, so this is a specific product of digestion of cross-linked fibrin generated via thrombin and factor 13A and plasmin activity. So this is the marker of thrombosis and fibrinolysis. Okay? So, to measure the fibrinolytic activity, ang mini-measure po natin is the D dimer. Okay. So, this is a schematic diagram po for you to visualize yung role po ng mga ating enzymes. Okay. So, we have plasminogen. Okay. Plasminogen, in the presence of tissue plasminogen activator, it will be converted to plasmin. Okay? So, ang inhibitor in this part is we have the plasminogen activator inhibitor. So, siya yung humaharang sa function ni TPA. Okay? So, once it becomes, what do you call this? Once it becomes plasmin naman, okay? So, your plasmin po, it will act in the lysine or arginine portion ng inyong fibrin. Okay? So, si plasmin, kakapit siya doon, it will initiate your degradation. Okay? So, ang function ni antiplasmin naman is to counteract that effect. Okay? So, once po si antiplasmin ay nakakapit na kay fibrin, okay, is magiging, what do you call this, resistant na siya sa effect ni plasmin. So here we have another one. Okay, so we have fibrin. So it has lysine or arginine terminal ends. Okay. Yeah. So we have taffy naman. Si taffy po, okay. So si taffy, ang role naman niya is it also works on this portion. Okay. So tinatanggal niya yung lysine component. Okay. Or tinatanggal niya to kasi dito kumakapit si plasmin, di ba? So kapag wala na to, walang kakapitan si plasmin, so hindi madedegrade yung inyong fibrin clot. Okay, so we have, let's skip that one. Let's proceed with this one. Okay? So if you remember this, ito yung itsura ng fibrin clot natin, di ba? Ang fibrinogen natin merong DED na portion. Okay? And then magdidikit-dikit sila. Okay? And then itong cross-linking na to between the both D is the effect of your factor 13. Okay? With stabilizing factor. Okay? So ito na yung fibrin clot mo pag magkakadikit-dikit na sila. Okay? In the presence of plasmin, it will fragment into different types. Fragment X, when we say fragment X, magkadikit pa C, B, E, and D na molecule. Okay. When we say fragment Y naman, okay, so fragment Y consists of D tsaka E lang, nawala na yung other D na domain. Okay? And then when you say fragments D and E, sabihin magkakahiwalay na talaga silang tatlo. Okay? Then when you say DED naman, DED is tatlo nga sila pero iba yung placement. Okay? Iba yung placement or pagkakadikit ni E domain kaysa dun sa dalawang D, and then yung D dimer or the DD is yung magkadikit na D. Ayan. So, bakit po si D-dimer yung ating mini-measure? Okay? Kasi, okay, yung D-dimer mo, it shows that you have a successful blood clot formation. Okay? Why? Kasi B and D is magkadikit sila because of the linking or cross-linking na effect ni factor 13. Okay? So, di ba si plasmin? Your plasmin works on both the fibrinogen and the fibrin clot. Okay? So, when your plasmin degrades fibrinogen, it won't have your D-dimer. Okay? So, ibig sabihin, na-lyse po siya even without successful blood clot. Okay? And then, kapag meron kang D-dimer, okay, since D-dimer is formed due to the effect na ni factor 13A, meaning you already had a stable clot, and then tsaka siya na-degrade. Okay? So, kaya po ang minimeasure natin usually is si D-dimer. Okay? Okay. Next, we have other inhibitors. So, we have protein C and protein S. Okay? So, this is already mentioned in the secondary hemostasis. Okay? Protein C is a complex with protein S and inhibits factor 5 and 8. It inactivates inhibitors of plasminogen activators. Okay. So protein C and protein S are cofactors. It enhances fibrinolysis by inactivating inhibitors of plasminogen activator. We also have antithrombin, okay, or formerly known as antithrombin 3. So this one inhibits plasmin and kallikrein, okay, synthesized in the liver and enhanced by heparin. So inhibits plasmin and kallikrein. Yes. So si plasmin, so we know the function of the plasmin, si kallikrein. If you forgot na, si kallikrein is involved in the intrinsic pathway. Okay? So si antithrombin, it hinders the activation of your intrinsic pathway. Okay, we also have what we call C1 inactivator. So this one also inhibits plasmin. And okay, so key takeaway from the discussion. So maikli lang naman ang fibrinolysis. So madali lang tandaan kasi mas konti yung mga involved. Okay? So the plasminogen and tissue plasminogen activator are absorbed within the formed clot. So the molecules are readily absorbed in the clot due to their high attraction or affinity with fibrin. So the plasminogen within the clot is transformed into plasmin that has the capability to break fibrin clots into fragments. The conversion of plasminogen to plasmin is also initiated by several components such as urokinase, activated protein C, and contact activators. So how do we test for fibrin degradation products or fibrin split products? Okay, so we can use either fibrin degradation product or FDP. So this test detects the late degradation products, fragments D and E, and but not the early ones, fragment X and Y. Okay, so di ba may hierarchy tayo, X muna, then Y, and then bago yung hiwa-hiwalay na D and E. Okay? So D-dimer assay naman evaluates fibrin degradation. So it is a non-specific screening test that is increased in many conditions in which fibrinolysis is increased, such as DIC and fibrinolytic therapy. Okay? Yan. So si DIC in the sense is disseminated intravascular coagulation, sobrang daming clot na namumuo. Okay? So clot ng clot sa katawan, kaya marami ding fibrin degradation products. Kasi clot ng clot sa katawan and then nade-degrade, dumadami si D-dimer natin tinatawag. Okay, so we will further discuss the diseases pagdating po natin sa diseases. Okay. So aside from, as we have talked about, okay, so other functions of plasmin aside from degradation ng fibrin and fibrinogen, it destroys clotting factors 5, 8, 9, and 11. Okay? So, it stops or it plays a factor in stopping the coagulation cascade. Okay? So aside from pagtunaw ng inyong final product, it also plays a role in stopping or slowing down the coagulation. Indirectly enhances or amplifies conversion of factor 12 to factor 12A, enhances or amplifies conversion of pre-kallikrein to kallikrein. Plasmin also promotes coagulation and activates the kinase and complement system and cleaves C3 protein of the complement system into fragment. Okay, so that's it for our discussion. I hope you have learned something. If you have any questions, feel free to message me. Okay, so thank you for listening and have a nice day.